All right now it's just me, so hopefully somebody else pops in who can come to the review. If not, I'm going to go through everything like I have in the past. Unit 3 is on reproduction, so it's going to be on mitosis, meiosis, and genetics, and I have a list of slides built out based on what people requested in the last lecture quiz. So let's get into it. For Unit 3, these are just some of the major overviews for chapters. Chapter 8 was mitosis and cytokinesis, the cell cycle, and cancer. So what happens when the cell cycle isn't controlled, and we'll talk a little bit about that today briefly. And then you have meiosis 1 and 2 in chapter 9, comparing mitosis to meiosis, which means comparing sexual and asexual reproduction, as well as talking about non-disjunction and what happens when meiosis goes wrong. And then chapter 10 is genetics with Punnett squares, non-Mendelian genetics. So Mendelian genetics are the very easy 2 traits. You're either tall or short. You're either green or yellow. And then the non-Mendelian genetics would be the weird ones, as well as pedigrees and vocab. So many of the concepts that I had questions on were more in the genetics, but I do want to review mitosis and meiosis to make sure that we're good. So let's start off with the cell cycle and mitosis. In mitosis, one of the big things that I want you guys to be aware of and be prepared for during the exam would be if you are looking at an image, how do you tell if it's mitosis or meiosis? And one of the things that you need to be able to do is tell me if I show you a picture of a slide and I say, or an image like this, if I say, is this mitosis or is this meiosis? I would want you to be able to tell me it's mitosis because it's a singular cell division that ends in 2 identical daughter cells, and it goes through PMAT once, which would be that prophase, metaphase, anaphase, and telophase. And we know that this is a animal cell because it does not have a cell wall, and then it went through cytokinesis using a cleavage furrow because it's an animal cell. So we also would know that this is mitosis because you get a single line of all of the chromosomes. So in this case, 4 chromosomes, you get a single line of all 4 chromosomes. And the sister chromatids separate during anaphase, leaving us with 2 new nuclei that still have 4 chromosomes apiece, which makes them diploid or having 2 copies of each chromosome. So mitosis should be pretty, hopefully feeling good with that. And then I did want to talk briefly about the cell cycle and what happens in the cell cycle checkpoints and the different stages. So the cell is in interphase for about 90% of its life. Interphase is often mistaken as the cell resting, absolutely not resting. And you can see that in G1, the cell is going through nutrients growth, checking its DNA, and it has a checkpoint in G1 that says, are you good to go? And if yes, then it will move into the DNA synthesis stage of interphase. The S phase or DNA synthesis stage is where we do see the DNA get duplicated in order for it to move into mitosis or meiosis. And then there's a checkpoint in G2 that is making sure that the cell is the right size and that the DNA replication was correct. DNA makes mistakes all of the time. We're going to talk about those mutations in chapter 11, but understand that when you proofread a paper, you're going to catch spelling mistakes, simple things like inverting the H and E and the and writing te, absolutely a thing that happens in DNA. So it gets checked in that G2 checkpoint. If it is all good to go, it will either go through mitosis or meiosis depending on what type of cell it is. If it's a body cell, it'll do mitosis. If it is a sex cell or germ line cell, it will do meiosis. Either way, they're doing these checkpoints and it has interphase. There is also a step during metaphase to make sure that the spindle fibers attach to the chromosomes and that everything is lined up appropriately and separates appropriately. If it's a body cell that has these mutations that allow it to skip the checkpoints, it might become cancerous. So if it skips the checkpoints and keeps dividing out of control, it's cancer, and that's just a disease of the cell cycle. If that cancer cell moves throughout the body and metastasizes, then it becomes kind of a big problem. Sometimes we see tumors form, and those are benign. Either way, we would treat that person as if they had cancer to make sure that they were taken care of appropriately. If the cell goes through cell division without passing the checkpoints properly, it has damage to the DNA, it has mutations, it has things that are wrong, and it gets caught by the body. It might have divided out of control once or twice, but the body will catch that a lot of the times, and it will then tell those cells to go into something called programmed cell death, also known as apoptosis, and that's going to be due to an error or the cell function not working appropriately. So for instance, when your stomach cells replace themselves, it's very common for stomach cells to get damaged by the acid in your stomach. Those cells are consistently going through apoptosis. That's a very normal thing. Apoptosis is normal. Cancer is not normal when that cell is going through an abnormal cell cycle. And that's the majority of chapter 8, which brings us to a little bit of comparing mitosis and meiosis. So we should be fairly familiar with mitosis. Let's fill in some of the things we know. That we end up with 2 identical daughter cells, and our cells are diploid, or 2 copies of each chromosome. Okay, mitosis goes through PMAT once, and mitosis is a type of asexual reproduction, which can be used for growth and healing as well. That's what we use it for, but some animals and plants absolutely make copies of themselves asexually. If you haven't been following Charlotte the stingray, you should go check her out. There's a stingray in North Carolina that is pregnant, meaning she's carrying eggs and pups, but we don't know how, if it's whether asexual reproduction through something called parthenogenesis or whether she's having a hybrid. My bet is for parthenogenesis. Those are a couple of the major things of mitosis. We could put those 4. And then I like to always go to meiosis and see if I can do a direct comparison. You guys have done this a couple of times now, but if I know there are 2 identical daughter cells in mitosis, I'm getting 4 unique gametes in meiosis, which are the types of daughter cells. These are all haploid, so they're going to be N, not 2N, half the number of chromosomes. It goes through PMAT twice, and this is sexual reproduction, which means we get genetic variation here, and that is through crossing over and independent assortment, as well as fertilization. In addition, we might put something here that says body cells, right? And these are germ cells. All I'm doing is saying if I can put something in mitosis, I need to be able to put the opposite of that in meiosis. And if you have a comparison question on the exam, you have to give me both mitosis does this and meiosis does that to earn full credits on that. Okay. If you just say mitosis has 2 identical daughter cells, but you don't say meiosis has 4 unique gametes, then you only earn half credit. In addition, make sure you can tell me what they have that's the same. They both go through the cell cycle. They both have cytokinesis, right? They both have interphase. There's DNA synthesis, right? Something like that. Checkpoints. Either way, okay, you're going to put those in there. So hopefully you guys have looked at that. A couple of people on the pre-lecture and the quiz that included this comparing gave me one part of the answer. Make sure you give me both. Okay. And then once again, we might have to do a visual comparison of mitosis and meiosis. And in this image, I have mitosis and meiosis on this side. Okay. And then we have a division here. Remember, everything unique that happens in meiosis happens during meiosis 1. This includes the crossing over and independent assortment. But if I want my last step to have 4 unique pieces of genetic material, 4 unique gametes, I need those to be unique starting from the very beginning. If I don't shuffle things up in the beginning, I'm not going to get a unique outcome at the end. So one of the biggest things is, again, I would see PMAT once with an end product of 2 identical daughter cells. If I see PMAT twice, I am definitely looking at meiosis. If I see during metaphase 1 a single line of chromatids, those duplicated chromosomes, I'm in mitosis. If I see homologous pairs lined up independently with crossing over, I'm absolutely in meiosis. If I see in anaphase my sister chromatids separate, but I have the full set of chromosomes, absolutely mitosis. If in anaphase 1 I see sister chromatids are not the ones separating, but my homologous pairs are, I am absolutely in meiosis. Or if I do see 2 cells with the chromatids separating, I am still in meiosis because I have 2 cells in meiosis too. And you would see 2 cells. I would never show you 1 cell and say, is this mitosis or meiosis? If I wanted it to be meiosis, you would see 2 cells. So make sure you go through those. I saw a few questions on that. I think the biggest questions with meiosis, though, is what happens that's different between meiosis 1 and meiosis 2. So one of the things I do want to point to would be some of the differences in these particular stages. Let's see if I can make this nice and big here. Hello, Rowan. So I have my camera off right now, but my cats are all coming to say hi. It's pretty fun. All right. In meiosis 1, this is where everything unique happens, okay? Prophase, we still see the nuclear envelope dissolve, right? Envelope. And it has to dissolve in the prophase 1 because if it doesn't dissolve, we can't move that genetic material around. It's a barrier, okay? And we see crossing over between paired homologs. So what this means, let me make this a little bit smaller here. What this means is that my homologous pairs, my sets of DNA that are given to me from mom and dad are going to find each other in prophase 1, and little segments of non-sister chromatids are going to exchange genetic information. We call it crossing over because the non-sister chromatids literally cross over top one another in order to exchange that genetic information. Then we see metaphase 1, the homologous pairs or homologs, either way, okay, align independently. So in this case, this particular cell has 6 chromosomes in its diploid state. So I will see 3 pairs of homologs in the middle because those homologs are finding each other. So if chromosome pair 1, there's 1A and 1B, 1A and 1B find each other, 2A and B, 3A and B, and then they line up down the middle of that cell. In anaphase 1, my homologs separate, so I am not separating the sister chromatids, okay? I am separating the homologs, and they are going to pull to opposite ends of the cell. And then in telophase, the nuclear envelope reforms. But my nuclei are now haploid, so half the number of chromosomes. So they're going to be 3 chromosomes per nuclei. Since I started with 6, I'm going to put here 6 chromosomes in my diploid cell. Okay. Cytokinesis occurs. It has to occur with meiosis, but it does not have to occur with mitosis. But in meiosis, the goal is to have always, always, always those 4 gametes. We're going to see meiosis occurring with cytokinesis. So once we have meiosis 1, we are going to end with 2 new cells that are haploid, and we move into meiosis 2. So again, I'm going to make this a little smaller and adjust this just a little bit. Make this a little bigger. We would see that in prophase, the nuclear envelope breaks down, but there's no crossing over, nothing crazy there. In metaphase, I'm going to have the chromosomes align, and these are duplicated chromosomes or sister chromatids. Let me put that in here. Sister chromatids align in the middle of the cell. In anaphase, the sister chromatids separate. And then in telophase 2 in cytokinesis, I get 4 new nuclei and 4 new cells. Remember, cytokinesis is division of the cytoplasm and organelles, where meiosis and mitosis is division of the nucleus. So telophase is going to be the nucleus reforming, because that's about the nucleus. Cytokinesis is the division of the cells. And that was one of the big ones that I had questions on. But I do want to talk how we get these unique individuals. So meiosis 1 and 2, the big thing, remember, meiosis 1 is everything unique, and the big 2 there are crossing over and independent assortment. So how do we get unique individuals was one of the quiz questions, and people didn't fully answer it. So let's answer it together so that we all have a rough idea of what that should look like, okay? So in prophase 1, crossing over exchanges genetic segments between non-sister chromatids of a homologous pair. This forms recombinant DNA. So I'm using vocab from that vocab list. Basically what we're doing is we are creating DNA that has not existed. That's why we call it recombinant, because it has recombined between the homologous pairs. That might be that if dad gave you a gene for brown eyes and mom gave you a gene for blue eyes, they might swap that so that there's now no new chromosomes that have all of dad's information except for mom's gene for blue eyes and vice versa. And that's creating new combinations of DNA, okay? In metaphase 1, homologous pairs align independently, which means that homolog pair 1 does not determine the arrangement of homolog pair 2, 3, 4, so on. I always liken this to if I tell people to go stand in 2 lines, 2 single file lines, and then I tell them to sit down, and I tell them to stand up and go line up in 2 single file lines and sit down and do it again and again. I'm going to get different combinations, but maybe think about it as if they have a partner and they have to line up with that partner. It doesn't matter whether the partner is on the left or the right hand line. They just have to be next to each other. Doesn't matter which order they line up in. Doesn't matter which order. That is independent assortment. So these 2 prepare the 4 unique gametes, okay? These unique gametes have not existed before. They're now haploid after going fully through meiosis. And then 2 unique gametes coming together in fertilization results in a new genetically unique or genetically, you are genetically different from me, okay? We would call that a zygote. So sperm and an egg come together. They're always going to be unique because the sperm and the egg are unique. And then which sperm and which egg actually go through the fertilization event is random. And so these are all random, which result in individuals with different DNA fingerprints. And if you think about it, that makes sense, right? That's how we can use DNA analysis to determine who maybe did a crime. That has its own problems. But it's also how we can say, okay, these are identical twins, or these are fraternal twins, or we know these people are siblings. It's how 23andMe works. So I want you guys to think through, if you have a question like that, crossing over independent assortment and fertilization, they're all random events that shuffle up the DNA and result in unique individuals. But if we're talking about it at the very in-depth answer, this is the most in-depth, most correct answer. Okay? And then let's briefly discuss non-disjunction, and then we get to pop into genetics. So chapters 8 and 9, mitosis, meiosis, you guys have been giving me fantastic answers. Make sure you review the homework and the quizzes and the vocab for those. But I think for the most part, everyone's pretty solid on those. most of the questions on the quiz were about about genetics. So briefly though, non-disjunction is a failure to separate. So in general, non-disjunction means failure to separate. And that is going to be in either anaphase 1, where the homologs fail to separate, or anaphase 2, where the sister chromatids fail to separate. In either case, if a cell is going to have a failure to separate, you will have a resulting gamete, okay? The resulting gametes is either n plus 1, so haploid plus an extra chromosome, or n minus 1, haploid minus chromosome. If that gets fertilized, so if we think about it this way, right, the sperm is maybe n, right, so a normal sperm fertilizes a non-disjunction egg, which would be n plus 1, right, you will end up with a zygote, okay, that is going to be 2n plus 1. 2n plus 1 would be things like trisomy. We talked about trisomy 21. In individuals that have Down syndrome, they have a third copy of chromosome number 21. You can have X chromosomes that are extra. My favorite X chromosome is all tortoiseshell and calico cats are female in that they are XX. But occasionally you will very rarely get a tortie or calico male. That individual has to be an event of non-disjunction because they have to inherit 2 recessive alleles to get the calico trait, and those are on the X chromosome, but they also inherit the Y for maleness, and so all tortie or calico male cats are XXY, but they are homologous recessive or homozygous recessive for the T or tortoiseshell color on the X chromosome. So there's just a fun little fact there as far as like calico cats are concerned. My outside cat, mama cat, is calico, so I know she carries that recessive allele because she has it, okay? All right. Important vocab. I pulled these directly from the vocab list. Now, should you know some other ones that I used in sentences? Absolutely. Things like recombinant, things like zygote, things like obviously, you know, sister chromatids and non-sister chromatids. But if I were to pull the most important vocab, this is absolutely the most important vocab. At the very bare minimum, you need to be able to use these in sentences and recognize what they mean in sentences. So if you haven't gone through vocab, definitely focus on these ones. Make sure you know them. Make sure you can define them. If you can't, look in your book. Your book has a glossary that defines all of these. Okay. These are the big ones for genetics too. So let's talk a little bit about genetics because I've had some people ask some questions and I want to clarify a couple of things here. So in genetics, okay, if we talk about Mendel and his P's, so these are Mendelian genetics. Mendelian genetics are the very straightforward sorts of genetics where I have a parental generation, so my P equals parental or purebred. So if these are my P's, for instance, I know that my P's are either white or purple, and if I let them self-fertilize, they're always going to white or purple depending on what the parent was. But if I cross those P's, my first generation is my F1 generation. And my F1 generation in this instance are all purple, okay? Which means my F1 generation inherits the dominant allele from the purple lineage and the recessive allele from the white lineage, making them heterozygous, okay? That's one of those vocab words. Heterozygous individuals have one of each type of allele in Mendelian genetics. Okay? If I allow that first generation, all of those plants are purple. If I allow those purple plants to fertilize themselves, I get an F2 generation. And my F2 generation, I'm going to have a different outcome. For some reason, 75% are purple, and 25% are white. Okay, I know that there's two potential genetic genotypes for purple. Okay, purple being my phenotype. Having the purple phenotype means that I have either the homozygous dominant genotype or I have the heterozygous genotype. Either way, I have a dominant allele, making me a purple flower. If I talk about the white flowers, all of my white flowers have to be recessive, homozygous recessive, because the only way you get white is if you have two recessive alleles. Okay, so what does that look like in a Punnett square? Well, let's look at it in a Punnett square, a normal Mendelian genetics Punnett square, where you have one trait with two different characteristics. So one trait being, in this instance, let's talk about eye color. And if I say eye color, okay, can be either brown or blue. That's it, okay, which isn't the case, obviously, but let's keep it simple. I know that there are two characters for that one trait. So when I want to do a Punnett square, I'm going to say let's cross two heterozygous individuals, meaning these individuals have the recessive allele. They're going to be capital B for the first gamete is going to inherit a dominant allele, and the second gamete will inherit a recessive allele. And then the same thing for the second parent. They have the ability to donate two different alleles. Okay. When I cross my Punnett square, which I think you all did a great job on the Punnett squares, on a Mendelian genetics Punnett square, always just use the same letter, very simple. We are going to see this outcome where I end up with one individual who is homozygous dominant, two individuals who are heterozygous, and one individual who's homozygous recessive. Now, I always put the dominant allele first because if you think the capital letter should go before the lowercase letter, that's perfect. Plus, I could, if I were keeping it in order, separate it and put it that way, but that makes me think that I have 4 different genotypes when in reality I have 3. So the other thing would be to always write, if I ask you for a genotypic ratio, write it out like this, where I can go, oh, okay, I have one individual who's homozygous dominant, 2 capital B's, right? 2 individuals who are heterozygous, 2 capital B and a lowercase b, and 1 individual who is homozygous recessive, which gives me a 1 to 2 to 1 genotypic ratio where I'm literally looking at the genes possible in those individuals. And then my phenotypic ratio, if I know particularly I'm looking at brown and blue eyes, I know that 3 individuals have brown eyes and 1 individual has blue eyes because I was told that brown is dominant to blue. And in my cross, if they carry the dominant allele, they have that dominant allele. They cannot have blue eyes. They can only have brown. That's a very normal Mendelian Punnett square. You will be asked to do a Punnett square on the test. If I want a normal Punnett square, I will just say Punnett square, okay? Do not look at it and say, oh, she's asking for one of the weird ones, because I know a couple people asked me about that. Absolutely not, okay? But let's take this moment to talk a little bit about genotype to phenotype. So the genotype is the genes possible. So that's going to be the individuals if they're homozygous dominant. Okay, in this case, having 2 dominant, 2 capital letters, heterozygous or homozygous recessive. Okay. And when I talk about the phenotype, I mean the physical characteristic. And this is going to be either dominant, in this case brown, or recessive, in this case blue. Okay. So if I ask the question, true or false, all individuals with the same genotype have the same phenotype, 1, you should always be wary of anything that says all or none, because in science, that is never the case. It's never all or none. But if I say all individuals with the same genotype have the same phenotype, that's true, because if you have the homozygous dominant genotype, you're going to have the dominant phenotype, okay? But if I say all individuals with the same phenotype have the same genotype, that isn't always true, because you could be either homozygous dominant or heterozygous and share the same phenotype. Now, if you have blue eyes like me, you know that you have the same genotype as anybody else with blue eyes because it's a recessive trait. But if you have brown eyes, you don't know if you are homozygous dominant or heterozygous. All you know is you have the dominant physical characteristic or the dominant phenotype. Okay? All right. I have two more things that were kind of requested, and I do want to talk about a pedigree really quick too, just to make sure we're all on the same page. And then I think that was everything on my list. So let's talk a little bit about the non-Mendelian genetics in terms of the codominance and incomplete dominance and the sex-linked Punnett squares. So non-Mendelian genetics are the genetics that don't follow Mendel's laws of independent assortment or don't follow the two traits the two characters for one trait, like purple and white for flowers. Flowers are kind of weird. So let's use flowers as our example for the non-Mendelian genetics. I'm also going to talk about blood typing because someone asked me about that. So let's start with codominance. Codominance, if you remember, co means shared or same. So this is shared dominant trait, okay? This would be your AB blood type because your blood type is IA. It can be IB or it can be lowercase I, where these A's and B's actually stand for the different characteristic, the different expression of a antigen or a protein. So for instance, someone who has AB blood type, okay, we have ABO blood in humans. Someone who has AB blood type has a dominant A and a dominant B. You still only get two alleles, okay, but you inherit two dominant alleles rather than inheriting a dominant and recessive. An individual with O blood type has two recessive alleles, okay? So let's do that Punnett square really quick. I have type AB blood, okay? So I like to use myself as an example here. So I know that I have one allele that is going to be A and one allele that's going to be B. And if I were to have a baby, I could donate them a or a B. That is what they would get from me. They would get A or B. My husband is O blood type. Okay, whoops, lowercase, don't capital for that for me, thank you. My husband is O blood type, okay? I'm gonna delete this one here really quick. No, we're gonna just leave it, okay? This means that all of my babies, if I were to have a baby with my husband, this first baby would inherit the dominant A and would get a recessive I, which is trying to be superscript, and I don't want it to be. Here, I'll just copy and paste the I. Okay. Okay. And so would this baby, because if I look, right, I would donate an A, he would donate the recessive trait that doesn't have anything. And so we would have one baby that is fully A blood type, but they are a carrier for O. So I could have a grandchild with O blood type, but I could not have a baby with O blood type. Okay. There was some major confusion on this one on the quiz. So if you're not understanding that, go look at that again. Go watch the Amoeba Sisters videos on that. Blood typing is done by taking your blood and seeing whether or not it's reactive to the different antigens. So for instance, if you took my blood and you mixed it with people with A blood, I wouldn't have a reaction. If you mixed it with B blood, I wouldn't have a reaction. And if you mixed it with O blood, I wouldn't have a reaction, which means that I am AB. If you took my husband's blood and mixed it with A, it would clot. B, it would clot. O, it would not clot, which tells us that he's O blood type. Okay. Now let's look at this in terms of if I was looking at flower color. Okay. If I were looking at flower color and codominance, I would see red and white showing up as striped instead of showing as pink, right? Because red and white would end up being equally expressed. So if I had a individual who had, let's say CR, right? CW, so they carry, right? If I crossed two individuals that were red and white striped, I would see those individuals having 2 babies who are heterozygous, so they would be red and white striped, or 2 babies that are red, 2 babies that are white. Okay, codominance is both are equally expressed in the heterozygote. Okay, 2 dominants equally expressed in a dominant heterozygote. All right. However, if I were looking at incomplete dominance, it's a little bit different. Okay, incomplete dominance is also known as blending. So heterozygotes are blended. They're an intermediate between the dominant and recessive because the dominant doesn't fully mask the recessive. So in plants, this would be when red and white made pink. And if you wanted to, you could do a very simple Punnett square. If I were saying that I were crossing an individual who is homozygous dominant for red and homozygous recessive for white, okay, I would end up with all of my individuals being pink, okay, because they'd all be heterozygous. Or if I crossed 2 heterozygous individuals, right, I would see a different genotypic ratio. So here's the cool thing. If I had a genotypic ratio here, it would be 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. That's not any differently. But my phenotypic ratio here would be 1 red to 2 pink to 1 white. Okay? So incomplete dominance and codominance are kind of opposites of each other. You might see a question about them. If I wanted you to do a Punnett square, though, I would tell you in an incomplete dominance situation or in codominance, if I just say a Punnett square, this is what I mean. Remember that. And the same thing with sex-linked Punnett squares. So if I'm looking at a sex-linked Punnett square, I'm going to have to include the X's and the Y's because X and Y are important. So if I want to cross a color normal male, so I'm saying crossing a male that is not colorblind, so he's X normal Y, okay? And if you're asked to do this and you don't want to do the superscripts, that's fine. But I'm not going to put the Y there because he would donate an X to one gamete and a Y to the other. And the female would be, we're saying a carrier, so she would donate 1 X with the dominant allele, 1 X with the recessive allele. They would have 1 daughter who is homozygous dominant for color normal, 1 daughter who is a carrier for color blindness or heterozygous, 1 son who is color normal, and 1 son who is color blind. Okay. Again, make sure that you're just tracking. If I want a weird Punnett square, I will ask you for it. And make sure that we're tracking that males only inherit 1 X. So if it's a sex-linked thing, they are going to always have the trait. Whether it's dominant or recessive, they get 1, that's what they get. Daughters can be homozygous dominant, heterozygous, or homozygous recessive. Males are just dominant or recessive. So X with a lowercase n and a Y is going to be recessive male. In order for a daughter to be recessive or to have that color blindness, she has to inherit 2 recessive traits. Okay? And then last thing is I want you guys to look really quick at a pedigree. You guys did really good on this on the quiz. So I just want to show us one just to make sure that we are tracking that. I'm going to adjust the slide. So if I have a picture like this, this is one that is showing a little bit more. This is a sex-linked trait. Again, all females are circles, all males are squares. My first generation would be this top 2 individuals here. My second generation would be the next line. Third generation would be the third line. Fourth generation, the fourth line. And we count individuals across to denote their numbers. So individual 1-1 is the female who starts generation 1. And we know that she is a carrier because she will be X capital HX, lowercase h, if she's carrying hemophilia. And she married a man who does not have hemophilia, and so he is not, he's just a square, okay? In their children, they had 4 children. They had a son who is a hemophiliac. Okay, their first son has hemophilia. He's a dark male, and they're showing you that on here. And he married a normal female, and they had 2 sons without hemophilia because she's not a carrier. She's homozygous dominant, and they had a daughter who's a carrier. In this second generation, his sister is also a carrier like their mom. She married a male that's a normal male. They had 3 children who were not hemophiliacs. A hemophiliac married into the family, and then they had children who were hemophiliacs, including a daughter that was hemophiliac. Let me find another one really quick. It's maybe a little less funky. One more pedigree here. This one has the numbers to it, so I can say that if you tell me something about individual 1, 2, we would say that that is a female that has the trait that I'm looking for. And looking at this, I look at each generation that comes from this family lineage, starting with generation 1. Their 4 children, 2 of them had the trait, and in their children, they had the trait. This tells me that this is a dominant allele in that it is found in every single generation, where our hemophilia gets skipped generations as long as you're not marrying a hemophiliac. So in this instance, I know this is a dominant trait, and it's because every single generation has individuals, and you either have it or you don't. This could be something like Huntington's. This could be something like extra digits. Having extra digits is dominant to having 5 fingers or toes. And if we were talking, we can even do this with cats. If we were saying these were cats that were polydactyl, the female cat was polydactyl. She had 2 children that had extra digits. Those 2 children also had children that had extra digits, which makes sense because it's dominant. Okay, I think that's it. Most people had questions more on the Punnett squares and kind of the oddness there. Hopefully I went through that. If you're in doubt, if ever you're in doubt, go watch those Amoeba Sisters videos. They have a whole heredity playlist that is absolutely fantastic. I cannot recommend it enough. The test opens tonight at 11:59. You have until tomorrow at 11:59 to take it. Study hard, do really well, and if you have questions, send me an email and let me know. I hope you guys have a great rest of your night.